Hello students, welcome to N's Biology. Previously, we have discussed about the female reproductive part of the flower, that is gynoecium or megasporangium, and the structure of ovule and different types of ovule. Today, we are going to study the Megasporogenesis, that is the formation of megaspores from the megaspore mother cells, and also about the female gametophyte, that is the Embryosac. Let's start study. Dear student, Megasporogenesis. Mega means large. Mega means large. Sporo means spore. Genesis means generation or formation. Okay. Large spore generation. What is the large spore? The large spore, large is the female. Always the female spore is large in size in comparison to the male spore. That's why male spore is known as microspore. Microspore. Where the female spore is known as megaspore. Okay. We have already discussed it in the previous class. So, Megasporogenesis means, mega means large or female, sporo means spore, genesis means generation or formation. Formation of megaspores from the megaspore mother cell is known as Megasporogenesis. The process of formation of megaspores from megaspore mother cell or MMC, M for mega, megaspore, M for mother, C for cell, megaspore mother cell, MMC, which is diploid in nature. So, the process of formation of megaspore from megaspore mother cell is called as Megasporogenesis. Okay. The formation of megaspores or the female spores from the megaspore mother cell is known as Megasporogenesis. Okay. So, which is the megaspore mother cells? We have already discussed about the structure of ovule in the previous class. Okay. Suppose this is an ovule. Okay. And here this and this is the integument. This is inner integument and this is outer integument, which cover the body of nucellus. This is nucellus. This part is nucellus, which is compactly arranged with parenchymatous cells, which is fill in reserve food. Fill in reserve food. Okay. Which we have already discussed. Okay. And this is the funicle. The point where the ovule is attached to the placenta or the ovary wall is known as funicle. And this part is hilum, where the body of ovule attached to the funicle is known as hilum. Okay. And this part, the swollen part, is known as chalazal pole. This part or this point or this pole is known as chalazal pole, and this part is known as chalaza. Chalaza. This part is known as chalazal pole and this point is known as chalaza. Okay. And where the integuments are absent, the opening portion is known as micropyle and the pole is known as micropylar pole or micropyle pole, which we have already discussed. Okay. Now we have to know which is the megaspore mother cells, from which cell the megaspore will be developed. Okay. This is nucellus. Okay. And the nucellus is compactly arranged or filled with many cells. Okay. Parenchymatous cells which are compactly arranged with each other. One of the cell of the nucellus towards the micropylar end. This is micropylar end. Micropyle. This is chalaja. Okay. Inside the nucellus, one of the cell which is present towards the micropyle is known as the megaspore mother cell. Okay. And which later developed to form the embryosac. It will be developed to form the embryosac. Okay. Embryosac or the female gametophyte. Female gametophyte. Okay. So this cell, out of total cells present inside the nucellus, one of the cell which present towards the micropyle is known as the or function as the megaspore mother cells, which enlarges in size and divide meiotically. Meiotically means meiosis divisions. Always the meiosis division occurs in the reproductive part. We have already discussed it. Okay. The megaspore mother cells get enlarges in size and divide meiotically. Let's take the megaspore mother cell here and draw here. Okay. Suppose this is one of the cell, that is megaspore mother cell or MMC. Okay. Here the megaspore mother cell is filled with dense cytoplasm. These are cytoplasm. Dense cytoplasm. And they are present a prominent nucleus. Nucleus. A prominent, a clear nucleus is present inside this cell, inside the megaspore mother cell. When the megaspore mother cells get enlarges in size, it undergoes meiosis division and produces megaspore tetrads. Tetrads means 4. Okay. In case of meiosis division, there 4 cells are developed from a single mother cells. Okay. Though here occur the meiosis divisions, there are formed 4 megaspores. Okay. Known as megaspore tetrads. Okay. Tetrad means 4. 1, 2, 3 and 4. Okay. Which are also filled with dense cytoplasm and they are present a single nucleus in each cell. Okay. The megaspore mother cell is diploid in nature. always the mother cell is diploid in nature. Okay, it contains 2n number of chromosome. Though it undergoes meiosis division by forming 4 cells, all cells are haploid in nature. Haploid means all are contains half number of chromosome, that is n. Here the mother cell contains 2n and the formed megaspores contains half number of chromosome, that is n, and all the 4 spores are haploid in nature. Okay, and the shape of the megaspore tetrad is linear. Okay, this arrangement is known as linear shape. Okay, this is one cell, this is second cell, this is third cell, and this is the fourth cell. The 4 cells are formed. So this is the total process of megasporogenesis. Okay, megasporogenesis means formation of megaspore tetrads from the megaspore mother cell. Which is the megaspore mother cells? One of the cells present inside the nucleus towards the micropylar end is known as megaspore mother cell. Clear? पिलामाने मेगास्पोरोजेनेसिस गोटे प्रोसेस जहा 4टा मेगास्पोर फॉर्म करूची फ्रॉम द मेगास्पोर मदर सेल्स. जेतबेले मेगास्पोर मदर सेल रु 4टा मेगास्पोर फॉर्मेशन होउची सेही प्रोसेस को कहा जाउची मेगास्पोरोजेनेसिस. ओके, एंड मेगास्पोरोजेनेसिस रे किए होउची मदर सेल्स मातृकोश कोउटा को कोषरु 4टा कोष मेगास्पोर क्रिएट होउची ना न्यूसेलस भितरे अनेक गुड़िए सेल कंपैक्टली अरेंज हेगी रहिचंती. सेगुडा को होचंती कोन ना पैरेनकाइमाटस सेल्स व्हिच आर फिल्ड विथ रिजर्व फूड मटेरियल. फूड मटेरियल रे भरी की रहिचंती जहा की जो एम्ब्रियोसैक डेवलप होउची बा फीमेल गैमेटोफाइट डेवलप होबो ता पई से माने खाद्य जोगेबे. ओके, तो जो सेल गुडा को रहिला ता भीतर गोटाए सेल जे कि माइक्रोपाइलर एंड आड़ को रहिची से गोटे सेल कोन होउची साइज रे बड़ हेका जाउची. जेतोले बड़ साइज रे हेई जाउची तो सेटा को कहआ जाउची कोन मेगास्पोर मदर सेल बा एमएमसी. M for megaspore, M for mother, C for cell, megaspore mother cells. And these megaspore mother cells undergoes meiosis division. ए mother cell टा कौन होउची meiosis division होउची, अर्ध विभाजन होउची. ओके, तो अर्ध विभाजन होइकरी कौन कहउची ना 4टा सेल क्रिएट करूची ताको कहा जाउची कोन मेगास्पोर टेट्राड. ओके, and the mother cell is diploid in nature which contains 2n number of chromosome, and the formed daughter cell or the megaspore tetrads each contains haploid number of chromosome. जो मेगास्पोर 4टा मेगास्पोर फॉर्मेशन होउची से माने समस्ते कोन करूचंती मातृकोश बा मदर सेल रो आधा नंबर क्रोमोसोम नउछ. एटा 2n थिबारु समस्ते nn नेकरी सृष्टि होउचंती. तो एटा हमर सरीला मेगास्पोरोजेनेसिस. दिस इज ऑल अबाउट द प्रोसेस ऑफ मेगास्पोरोजेनेसिस. लेट्स स्टडी अबाउट द फीमेल गैमेटोफाइट और द एम्ब्रियोसैक. हाउ द megaspores are developed to form the female gametophyte or the embryosac. Let's see. Female gametophyte or embryosac. Let's see the development of female gametophyte or embryosac from the megaspores. Okay, or the megaspore mother cell. we discussed that the megaspores tetrad are formed from the megaspore mother cells. Okay. According to P. Maheshwari, she classified the embryosac or the female gametophyte on the basis of number of megaspore nuclei participating in the embryosac. गोटे एम्ब्रियोसैक आमे देखिले कोन मेगास्पोर मदर सेल तो ये मेगास्पोर मदर सेल्स डिवाइड हैला मियोसिस है कि कोन हैला दिस इज मेगास्पोर मदर सेल्स इट अंडरगोज मियोसिस टू फॉर्म मेगास्पोर टेट्राड okay on the basis of the number of megaspore nuclei participating in the embryosac formation, the embryosac is of different type according to P. Maheshwari. आमे देखिले गोटे मेगास्पोर मदर सेल डिवाइड है कि चारिटा मेगास्पोर टेट्राड फॉर्मेशन कला जहा भितर गोटे गोटे न्यूक्लियस रहिले तो माहेश्वरी कहले according on the basis of number of cells or the nucleus जदि मेगास्पोर बा फीमेल गैमेटोफाइट फॉर्मेशन रे बा एम्ब्रियोसैक फॉर्मेशन रे गोटाए सेल गोटाए न्यूक्लियस पार्टिसिपेट करूची तो सेटा को कुहा गला कोन मोनोस्पोरिक जदि दुईटा सेल दुईटा न्यूक्लियस पार्टिसिपेट करूची दैट इज बाइस्पोरिक जदि चारिटा जाको सेल पार्टिसिपेट करूचंती फीमेल गैमेटोफाइट डेवलपमेंट रे बा एम्ब्रियोसैक डेवलपमेंट रे ताको जाउची कोन टेट्रास्पोरिक एम्ब्रियोसैक सो according to P. Maheshwari the female gametophyte or the embryosac is of तीन प्रकारर monosporic embryosac when only one spore or single spore is participating in the formation of female gametophyte that is known as monosporic embryosac and the example is polygonum type and oinothera. The monosporic embryosac is polygonum type or oinothera type. Approximately 80% of angiosperm, the female gametophyte is of monosporic type or polygonum type or oinothera type. 80% of angiosperm 80% angiosperm by flowering plant तार female gametophyte by embryosac होजी monosporic. Okay, only a single spore participating in the development of female gametophyte. And bisporic when two spores 1 and 2 spores are participating in the formation of female gametophyte that is known as bisporic embryosac and the example is allium and endymion. Okay, then the third type is tetrasporic embryosac. Tetra means 4 when all the 4 megaspores are participating in the formation of a female gametophyte or embryosac that is known as tetrasporic embryosac and the example is adoxa, plumbago, drusa and fritillaria. Okay, adoxa, plumbago, drusa and fritillaria, these are the example of tetrasporic embryosac. I said that the 80% of angiosperm form monosporic embryosac. Monosporic embryo sac or female gametophyte. Let's see how the monosporic embryo sac is developed. Okay, the monosporic embryo sac, also known as polygonum type. Polygonum. Okay, let's see how it developed. First, from the megaspore mother cell. Megaspore mother cell undergoes meiosis to form megaspore tetrad. 1, 2, 3 and 4 cells are formed, and all are haploid in nature. Contains N. The mother cell is diploid. Okay. In case of monosporic embryo sac development, only one cell is functional. This is functional cell. Only one cell is functional. Okay. And other 3 cells are degenerates out. Okay. जो 4टा सेल रहिला 4टा मेगास्पोर मध्यरु गोटाए मेगास्पोर फंक्शनल रहउची और बाकी 3टा डिजेनरेट हे जाउचंदी बा नष्ट हे जाउचंदी. को 3टा सेल व्हिच ऑफ द 3 cells are degenerates out? Suppose this is micropylar end, this is chalazal end. Okay, or this is chalazal end and this is micropylar end. Okay, the cells from the micropylar end, 3 cells, 1, 4, 3 and 2. Okay, from the micropylar end ru 3टा cell, they get degenerates out, and the one cell which is present in the chalazal part is the functional cell. Okay, or the functional nucleus. Okay, other 3 from the micropylar end are degenerates out. Degenerates. तो 3टा सेल डिजेनेरेट हे गला, गोटाए सेल जे कि चलाजल एंड रहिला सेटा फंक्शनल हे कि रहिला. एगुडा को सब नष्ट हे गले. ओके, एंड दीज फंक्शनल सेल और न्यूक्लियस undergoes mitotic division. Mitotic by mitosis division. In case of mitosis division, the chromosome numbers are same. It is known as equational division by samobibhajana. When the cell, the functional cell, undergoes mitotic division or mitosis division, the nucleus divided into form 2 nucleus, 1 and 2 nucleus. Only the karyokinesis occurs. Karyokinesis means only the nucleus is divides. Okay, the nucleus is dividing to form 2 nucleus, but no cytokinesis occurs. Means the cytoplasm does not divide here, only the nucleus divides to form 2 nuclei. Okay, so after the mitotic division, here forms 2 nucleus. It is the first mitotic division. Again, the 2 cells or the 2 nucleus again divided mitotically, that is second mitotically division, to form 4 each. First, this cell undergoes mitotic division to form 2 nucleus, and again the two nucleus undergoes mitotic division to form 2 and 2, 4, and these 4 cells again divided mitotically to form 8 cells or 8 nucleus. 8 nucleus. Okay. So how many mitotic divisions occurs in the formation of female gametophyte? First, second and third. 3. You have to remember this. 3 mitotic division occurs in formation of female gametophyte or embryosac. First, 1 nucleus divided into 2, second ray 2 nucleus divided into 4, and third ray 4 nucleus divided into 8. After the 8 nucleate stage, ଯେତେବେଳେ 8ଟା nucleus formation ହଉଛି, the 6 nucleus out of 8, 3 nucleus are moved towards the chalazal end, and 3 nucleus are moved towards the micropylar end, and 2 nucleus here present in the center by attaching with each other. Okay. When the nucleus divided into 8 nucleus stage in the monosporic embryosac development, out of 8 nucleus, 3 are moved towards the chalazal end, and 3 are moved towards the micropylar end, and rest 2 nucleus present in the center of the spore by attaching with each other. The nucleus then gets surrounded by the cytoplasm. Hence, here the cytokinesis is occurring now. Okay. The cytoplasm now divided. Here in the chalazal end, 3 nuclei are surrounded by cytoplasm, and in the micropylar end, these 3 are also surrounded by the cytoplasm. Okay, and these 2 which present in the center is surrounded by single cytoplasm, or these are known as polar nuclei. Polar nuclei which present in the center, and the cells present in the chalazal end is known as antipodal. Let's draw the diagram here clearly. See here, the out of 8 cells, 3 are moves towards the chalazal end, and 3 are moves toward the micropylar end, and 2 remain attached in the center of the cell. Okay, the cells or the nucleus which are moved towards the chalazal end, they are covered with cytoplasm, forming the antipodal cells. The 3 cells present in the chalazal end is known as the antipodal cells. Okay, and the 3 nuclei which are moved towards the micropylar end and get surrounded by the cytoplasm, forming the 2 synergids and 1 egg cells. Out of 3 cells which are present at the micropylar end, 2 cells are known as synergids. These 2, this and this is known as synergids, and the cell, the third cells present upper to the synergid is known as egg cell. Okay, and inside the synergid they are present finger-like projection known as filiform apparatus. Okay, and this is the polygonum type embryo sac. Okay, or the monosporic embryo sac. Okay, clear? The development of female gametophyte takes place from the megaspore. Okay, and the formation of megaspore tetrad from the megaspore mother cell is known as megasporogenesis. Okay, after megasporogenesis, P. Maheshwari states that there are three types of embryosac or female gametophyte: monosporic, bisporic, and tetrasporic. When a single spore undergoes the formation of female gametophyte or the embryo sac, then the embryo sac will be known as monosporic, and when two spores undergoes the formation of female gametophyte, it will be called as bisporic. When all the four spores, all the tetrad of megaspores undergoes the formation of female gametophyte, it will be known as tetrasporic. Okay, in case of 80% of angiosperm, there is present monosporic embryo sac. Okay, monosporic embryo sac also known as polygonum type. Polygonum type. Monosporic means single spore. Out of 4 spore, 3 are degenerate out. Which 3? The cells or the spores from the micropylar end. Micropylar end pakuru tinita cell 1, 2, 3 are degenerates out, disappear he jauchanti, and the rest cell or the spore present at the chalazal pole undergoes the development of female gametophyte, that is known as functional cell, and these functional cells undergoes mitotic division to form 2 nucleus, and then second mitotic divisions to form 4 nucleus, and the third mitotic division to form 8 nucleus. Totally 3 mitotic division occurs, or the functional cell undergoes 3 mitotic division to form 8 nucleus. Okay, and out of 8 nucleus, 3 are moves towards the chalazal end and 3 are moves to the micropylar end, and remaining 2 nucleus are present in the center by attach with each other. Okay, after moving towards the 2 pole, chalazal pole and the micropylar pole, the nucleus are surrounded by cytoplasm and forming the cells. The 3 cells present at the chalazal end are known as antipodal cells, and the nuclei present in the center, 2 nuclei, are known as polar nuclei, and the 3 present in the micropylar end, 2 are known as synergids and 1 is egg cells. Okay, inside the synergid they are present finger-like projection known as filiform apparatus. Okay, and the synergid disappear towards the development of male gametophyte or at the time of pollination. When the pollination will be occurred, then the synergids will be disappear. It only helps to the entry of the pollen tube. The pollen tube entry करिया ପାଇଁ कि synergid ଆଉ तार filiform apparatus help करूचंती. Okay. So the female gametophyte or the monosporic embryo sac is 8 nucleated and 7 cell structure. Okay. 8 nucleated. 8 nucleus. When someone asking you about the number of nucleus and number of cells of the female gametophyte, the answer will be it is 8 nucleated and 7 cell structure. The monosporic embryo sac or monosporic female gametophyte is 8 nucleated and 7 cell structure, that is polygonum type. 8 nucleated, how? 1, 2, 3. 3 at chalazal end, 3 at microbial end and 2 are at the center. So totally 8 nucleus or 8 nuclei are present and 7 cell. 1, 2, 3, 4, 5, 6, 7. 7 cells. Okay. It is all about the female gametophyte. In the next class, we will study about the pollination. Okay. Till then, keep watching and keep learning. Don't forget to subscribe the channel, and if you are liking the teaching process, don't forget to touch the like button and comment and share, and thank you.